What's up, biology students? Mr. Holloway here. We've been talking a lot lately about what DNA does inside of our cells. But what happens to that DNA when cells divide to make more cells? How do those new cells get the DNA they need in order to build proteins? In today's video, we're going to learn about DNA replication, the process used by cells prior to division to ensure that each new cell will get a full and complete set of genes. Remember, all cells come from other pre-existing cells. That's one of the main points of cell theory. So that means that every cell in our body, all 100 trillion of them, came from cells that already existed. More specifically, they all came from one cell, called a zygote, depicted here at the very beginning of our figure. This zygote is the product of sexual reproduction. Two cells, an egg and a sperm, from two different organisms of the same species, each containing half the DNA necessary to build a living organism. When these two cells combine during fertilization, the DNA from the sperm cell is added to the DNA inside the egg cell, forming a cell with a complete set of genes. Soon that zygote begins to divide, over and over again, forming an embryo. Eventually, the cells in that embryo begin to organize themselves into groups that will one day become organs, and the cells themselves begin to specialize and take on specific roles based on which genes inside get turned on and which genes get turned off. Every cell in your body today is derived from that one original cell, that zygote, and that zygote contained all the genes necessary for building you. And the cells that make up your body today still contain the same set of genes. A few random mutations aside, every cell in your body contains exactly the same DNA. Every time a cell divides, the DNA inside is carefully and meticulously copied to ensure that each new cell receives a full and complete set of genes. When this process goes wrong, it can have very serious consequences, especially if it happens during one of the embryonic stages of development pictured in this figure. In case it has not already been made clear, DNA is a pretty incredible molecule. The fact that this molecule contains information that can be read and interpreted by the cell in order to manufacture proteins is remarkable in and of itself. DNA can also replicate itself with assistance from a variety of enzymes, and it does so accurately and precisely almost every single time, which is pretty amazing considering the fact that human DNA contains over 3.5 billion base pairs. Imagine being asked to precisely copy down a code of 3.5 billion letters all in a row. Then imagine doing that over and over and over again without making any mistakes. If you can imagine that, you'll come close to recognizing how remarkable DNA replication really is. DNA is able to do this because of the rules of base pairing. When DNA is unzipped, as shown here in this image, enzymes simply grab nitrogenous bases that are floating around freely in the nucleus and match these up with the appropriate base on the pre-existing strand of DNA, A with T and G with C. I say simply, but it's actually quite a complex process. Because of the rules of base pairing, each strand of DNA in a double helix is essentially a template for building the other strand. During DNA replication, the DNA double helix is literally unzipped by enzymes, separating each strand from the other. Additional enzymes work to add new nucleotides opposite of each exposed strand of DNA, once again using the rules of base pairing. The end result is two identical strands of DNA, each containing one original strand and one new strand. Not only are these two new DNA molecules identical to each other, they are each identical to the original strand we started with. This process occurs just prior to cell division, and each new cell produced during cell division will inherit one of these replicated DNA molecules, and thus the genetic code is passed from one generation of cells to the next by way of asexual reproduction. Let's take a closer look at how this process plays out. Here we see a segment of DNA being replicated with the help of several important enzymes. Remember, enzymes are proteins, and the instructions for assembling these proteins are coded in our DNA. So it is worth noting that some of the genes in this molecule of DNA code for the enzymes needed to replicate this molecule of DNA, which is kind of a cool irony of life. This enzyme here is responsible for unzipping the DNA double helix, and it is called helicase. Different protein molecules bind to the DNA to hold the double helix apart here at the replication fork, and an enzyme called DNA polymerase lays down complementary nucleotides using the existing strand as a template and matching up bases according to the rules of base pairing. here's where things get a little tricky. Because in a DNA double helix, the two strands of DNA run in opposite directions. They are anti-parallel to one another. Although all the same basic rules apply to both strands of DNA, the way in which each new strand is built to match varies considerably. On the so-called leading strand, the new strand being built is synthesized continuously in one long chain working in this direction toward the replication fork. That's what we see on this top strand here in our diagram. On the other strand, the so-called lagging strand, the new strand being built is synthesized discontinuously, which is to say that it is built in fragments. These fragments are called Okazaki fragments, after the scientist who discovered this particular detail of DNA replication. First, an enzyme called RNA polymerase, you've actually heard of this one before, lays down an RNA primer, or starting block. This starting block is just something to work off of. It's basically the starting line for DNA replication on the lagging strand. Then, DNA polymerase enzymes build a short section of DNA, starting at the primer, and following the lagging strand in this direction. These RNA primers are broken down by DNA polymerase and replaced with DNA bases later on. Then an enzyme called DNA ligase knits everything together into one continuous strand. This fragment would have been the first one built. Then the enzymes jump backward toward the replication fork and build another short segment, once again following the lagging strand in this direction. When this fragment is complete, the enzymes jump backwards once again and build another segment, and so forth, following the replication fork just as the leading strand does. If we zoom in on this picture here, we can see that this process is actually happening in both directions, and that each side is essentially a flipped mirror image of the other. Whereas the leading strand is on top and the lagging strand is on the bottom going in this direction, the lagging strand is on top and the leading strand is on the bottom going in the other direction. Remember, you can always recognize the lagging strand because it is being built in fragments, and those fragments are called Okazaki fragments. As DNA helicase enzymes continue to unzip the DNA double helix in both directions, this replication bubble gets larger. The leading strands are still being built continuously, and the lagging strands are still being built in fragments, but the newest DNA is always going to be closest to the replication fork, going in both directions. These 5s and 3s help us to figure out direction and make some sense of these different styles of strand building. If this is where we started building the leading strand, we can see that we're following the existing strand from the 3' end to the 5' end. Same is true of the fragments on the lagging strand, and in the leading and lagging strands on the other side of the bubble. Because the two strands of DNA run anti-parallel to one another, and because we're moving from the 3' end to the 5' end of the existing strand of DNA, that means that new bases are added to the new chain from 5' to 3', which is what these numbers here are showing us. It's just another one of those rules, and one that I don't really care if you remember or not. But this rule about following the existing strand from 3' to 5' while building 5' to 3', and also while working towards the replication fork in both directions, helps us to understand why DNA is built continuously in some places and in these short fragments in other places. It also helps us to see just how complex and intricate this process is, and helps us to appreciate just how amazing the machinery of the cell really is, since it can do this accurately and correctly pretty much every time. During DNA replication, this is happening in numerous locations simultaneously, and a single DNA double helix will have many such replication bubbles scattered throughout this very, very long macromolecule. These replication bubbles eventually run into one another and merge, and once this has occurred along the entire length of a DNA molecule, we officially have two new DNA molecules, and DNA replication is complete. Keep in mind, in humans there are approximately 3.5 billion base pairs in our genetic code, and to replicate all that DNA, and to do it correctly and accurately every time, is a huge undertaking for the cell. But cells go through this process each and every time they prepare to divide. And it's a pretty darn good thing that it does happen correctly almost every time, because it has to happen millions of times in order for us to grow from a zygote into a fully grown human being, and continues to happen throughout our lives in order to replace cells that inevitably wear out and die. When it does go wrong, those mistakes, those mutations, are passed to every generation of cells that comes after. Which means that if it goes wrong very early in the existence of an organism, it could mean that most or all of the cells in that organism's mature body will contain this error. And with that, I will bring our video to a close. Thanks for watching, and feel free to go back and watch this video as many times as you need until you feel like you understand the process of DNA replication and how this process helps cells to pass their genes from one generation to the next.